India is surrounded by seas and oceans on three sides, and the world is having 70% of water. Marine, so we can imagine how much potential in marine drugs is available throughout the world. Marine organisms comprise of animals and plants, and they are the wealthiest source of bioactive compounds. Scientists have found out a lot of pharmacological activities from marine organisms lately, which is mainly in the areas of free radical scavenging activity, anti-tumor activity, antimicrobial, anti-inflammatory, antiviral, analgesic, neuroprotective, and immunomodulator activity. Now, recently, even anti-COVID activity has been reported from marine drugs. Now, very rightly, as Dr. Vijita said, it is in the year of 1969 that the first antiviral drug, that is cytarabine, was marketed, or it came in under clinicals. It was the first drug which was approved by FDA. It is famously also called as Akka, Arasi, or Cytosur-U. In the year 2019, the scientists had celebrated the 50th anniversary of this drug, and that was in 2069 that the first drug was marketed. After that, there has been not a huge variation, or there has been no huge findings in the marine field. After 1969, it was in 1976 that antiviral drug, which is used for treatment of herpes simplex, that is vidarabine, or it is also called as Ara-A or Vira-A, was found out, and it was marketed by FDA. Approval was obtained and it was marketed. So we are lacking back in marine drug discovery, and it is there is lots of myths about marine drugs, and the myth is mainly that you have to go deep into the ocean to get marine drugs. So it is not like that. You can also find it in fresh waters or cultivate marine drugs. So today's talk, I will be giving you new areas. I'm going to take you into new areas and try to be of some help for the researchers in this field. And I hope that I will do justice to my talk and to Dr. Vidyadhar sir, who has selected me for this talk. Today's contents will be concentrating on chemical classes and the sources of marine drugs, which is the most important ones. Then on marine pharmacology, I'll be talking only about the pharmacology of the drugs, which are important, of course, and the list of FDA approved drugs and the drugs which are in pipeline. What are the limitations of marine drugs? What is the meaning of aquaculture and how it is carried out? The simple extraction techniques of phytoconstituents from marines in labs, then systematic steps that you can do for the research of marine phytoconstituents, the names of the institutions and the research centers in India as well as in abroad who carry out a lot of research and who promote research in marine drugs. Then next is about molecular docking. I will also be throwing some light into what about the new drug, that is an antiviral drug, which has been identified from marine drug recently. Then I will be taking you on a visit through CMNPD database, which is mainly created for marine drugs and completely free of charge. Then what are the future prospects? Summary of the talk. We are all studying about marine drugs in our syllabus, especially M.Pharm pharmacognosy students, they'll be familiar with it. So I will not be going deep into it, but there will be other students also who would like to know what are the sources of marine drugs. So let me tell you, we can get marine drugs, as I said, from the seas, that is salt water, as well as from the fresh water. Marine sources are classified as fish, algae, porifera, Cnidaria, Mollusca, Chordata, Echinodermata, Arthropoda, fungi, and other microorganisms. So various types of antimicrobial drugs, anti-inflammatory drugs, anticoagulant drug, antihypertensive drugs, antivirals are isolated and studied upon from the marine source. And it is seen that marine drugs are having basically a toxic component. Now, this toxic component, even though it is toxic, it is not, all the marine drugs are not toxic to human beings, but because it contains the toxic component, even low doses of marine drugs can cause anti-cancer effect. So this is the biggest thing, and that's why there are lots of anti-cancer studies which have been carried out, especially on marine drugs. Now here I have got a diagrammatic view for you to understand. There are actually a lot of classes, as said earlier by madam. It is a biodiversity, because the best part is the marine drugs or the marine organisms, they grow in the deep oceans and seas where the environment is rough, and because they have to survive in that environment, they have themselves a mechanism of defense which is in their system, and this defense mechanism is containing a huge diversity of chemical compounds which are not available otherwise on land, or which we don't get from plants. Scientists have developed a lot of, they have synthesized a lot of drugs which mimic the same as that of the marine drugs. And you have low molecular marine phytoconstituents also, and high molecular also. But many are identified and they are having very good activity against vast number of diseases. So the main classes of chemical components, when we say they are classified as peptides, alkaloids, unsaturated fatty acids, imidazole, sterols, polyketidides, then polysaccharides, beta-lactam antibiotics, microcyclic lactones, and terpenoids, many. There are lots of important active constituents which are present in the marine drugs. In the next table, you can see I have given a little bit of classification, and this is all upon the important discoveries which have been come out recently. So one important antibacterial drug, that is iscosapentonic acid, has been found out, which is a polysaturated fatty acid. It has been isolated from Phodentylum trichonutum. The names are a little bit difficult to pronounce, but if you just see the history of the names, it will be very nice and interesting. It is mainly because of the color and the way it is, the marine drugs are very beautiful, and these marine components which are present in the seas and oceans, they are mainly containing the same type of cells. They are not very complicated in cellular structure, but each cells, or they contain a lot of chemical drugs which are found to be having huge bioactive nature. All right. Then another one, anti-inflammatory drug, has been isolated from Spongia officinalis, which has been found in abundance in India. Then a neuroprotective drug from Ulva reticulata, anti-herpes or anti-parasitic drug from the Sarcotagus species, which is also called as Tunisian sponge. And anti-cancer you have from many, but then FDA has now approved one drug recently, which is bryostatin from Bryozone. And the name of the organism is Bigula neritina. So this is just a little bit more I have focused on, so that you will come to know that you see many of the derivatives which are obtained are coded, or the coding is because they are under clinical trials. Okay, so there are three phases, as you know, phase 1, phase 2 and phase 3, and some are in pipeline also. So those drugs will not be named, they will be only having some code. So you get from various types. So I have listed out some more, like from sponges, actinomyces, mollusks, bryoza, even from shark. All right. So these are the names: AE941, bryostatin 1, dolastatin 10, and cahalidide F. Then cyanosporamide A, and various other drugs. These are the ones which are in pipeline, and the clinical trials are ongoing. Now, to have a vast view, because it will be useful, these are the vast varieties. I have focused on the important ones, but this table will give you a clear picture of which are the drugs which are approved. So you can see that there are 7 compounds, names are given, which are approved by FDA, and 3 are in phase 3. And there are around 7 more in phase 2, phase 1, and preclinical studies. That means that a lot of research is going on throughout the world. And in India also, there are marine centers, research centers, which have been opened and called for research work on marine drugs, is being highly focused on by the government of India. But there are also limitations. For every natural products, there will be limitations. But the limitations here are not the same as that is obtained in plants. Okay, here the limitations are that you will get very less number of active compounds, because a huge number of marine drug is marine source is required for isolating even a small amount. So because of that, there is lack of sustainable supply of original compound. And the taxonomy has been developed very much, and I will take I'll take a visit for you throughout that. So taxonomy is very well developed, yet sustainable supply of the original compound is found to be difficult. So if there is one step of research carried out, to repeat it or for advancement of the research, we are seeing a setback. So for this, to overcome, there are some techniques which has been developed. So increased development of synthetic and semi-synthetic analogs are going on. And how they are finding out the nature of the compound? That is by carrying out various biosynthetic studies. So by studying the pathways, the biosynthetic pathways are studied, and from that, using NMR studies and crystallographic studies, X-ray crystallographic studies, the structure of the compounds are analyzed. And then synthetic, you can get even synthetic drugs which are similar to that of the marine drugs. Then second limitation, which is a main limitation, is the structural complexity of isolated compound. Now these isolated compounds, most of the compounds have a very huge molecular structure. Now because of the huge molecular structure, first thing is isolation becomes a little complicated unless and until it is being carried out very carefully. And as I told you, the yield is little less. So because the yield is less and because of the structural complexity, the problem that is faced is there is wrong assessment. So even if you give for studies, the assessment is wrong. The compounds which are having a huge complexity will be attached to a various amount of groups and bonds, and due to which assessment, even by using the most sophisticated techniques, also becomes quite complicated. So for that, and to overcome it, nowadays there is a artificial culture that is being carried out. There are two types of culturing techniques, or cultivation techniques we can call it, which is done for marine drugs. It is called as mariculture and aquaculture. Now, mariculture is just like farming, and it is done in natural environment. That is, depending upon the conditions on which they grow. Under that database, a artificial pond is created, and or from the pond where it is obtained, or from the area where it is obtained, there it is grown in excess. It is done by farming technique, by providing nutrients which will aid in the growth of the marine origin or the marine source. So that is one way, and because it is carried out in natural environment, it is called as mariculture. The second one, which is in huge use, is called as the aquaculture. Now, aquaculture is where the culture takes place under artificial conditions. So I'll be showing some pictures related so you can come to know. And aquaculture is being practiced in India in many research centers, wherein they themselves grow it. They don't have to go and collect it. There is no loss in collection protocols. There is no loss in the chemical compositions, and a lot of benefit is there while doing aquaculture. So mostly all the researchers are nowadays following this technique, and that commercially now marine plants or marine organisms are obtained even commercially. So the commercialization has also started in a huge way, especially the sponges and the porifera. Many such type of important marine sources are being commercialized. Now, the third one is it is having a high hydrophobicity. What do you mean by hydrophobicity? Is it has, means when it is try to dissolve in water, or when you try to, when it comes in contact with the atmospheric moisture, then there is destruction in the composition of the components. Now, why this happens is because in the ocean, this usually takes place only for the sources which are obtained from the ocean, because deep in the ocean, there is a low oxygen content. And because of which, when it comes, it is adopted in such a way that it can grow in that condition. But that doesn't happen when it is being taken out. So high hydrophobicity is one main issue which has been faced, and now new methods or research also is being carried out to combat hydrophobicity of marine source. Now here in this slide, sorry, let's go back. Yeah. In this slide, you see both the methods, aquaculture, there is in situ method and ex situ method. Okay. So there are strengths, weaknesses, opportunities, and threats for both the methods. So I've just segregated it so that you can come to know, even for the researchers who want to do some entrepreneurship, or students who would like to do some entrepreneurship, because this is a vast growing area. It will be very helpful for them. So that's why I just thought I'll segregate it like that. So in situ method means what are the strengths? Strengths means benefits. Okay. So the benefits are that it is grown under natural conditions. So the original composition is not altered. The cost is very less, and the manpower required is also very less. So mainly algae, red algae, blue algae, green algae, fungi, all these can be grown by in situ aquaculture method. It is also very easily available in any ponds, and you might have studied about a lot of algae and fungi species which are used as medicine, spirulina and so many others. Okay. So being the strengths, what are the opportunities? The opportunities are that it has got, without spending much, you can carry out a business on it. So this business opportunity is very high for the drugs which can be obtained from in situ aquaculture method. But the weaknesses are that from area to area there can be variation, and because of this variation from area to area, you won't get the correct consistency, or the concentration of the components vary. So that is one main drawback which is seen in this method. And there is manipulation of culture condition, because it is the atmospheric conditions keep changing, so naturally the source, the plant, or the it's an animal or the microorganisms, they will manipulate among themselves, so there can be changes possible depending upon the culture conditions. Then genetic pollution will take place. That is because our fresh ponds or seas or oceans, they get polluted. So that is one another weakness. And it is little technically challenging. That is the collection and then the storage and everything is little bit technically it is challenging. Moreover, getting permission from the government is also a long process. Okay, so this is one major weaknesses that are found in this method. And what are the threats? The threats are that there can be natural disasters like storms, and because of that, the full thing that you have cultivated or grown, everything can get destroyed. Then the presence of diseases, then increasing limitation to the harvest of wild population. So when you grow this, what happens is many different types of fishes or frogs and other pests, they come and they invade, and that is another one threat. Then it can be easily trespassed, means it can be easily acquired by some other person and you will not be able to control it. Okay, so these are the 4 main points which are there in situ aquaculture method. And when you check it, when you compare both, you can see that in ex situ aquaculture method, the points of weaknesses and threats are very less. All right. So from this itself, you can understand, obviously, you can understand that ex situ aquaculture method is more beneficial. So now let us concentrate on the strengths. So whatever are the weaknesses, this side has turned into the strengths of ex situ aquaculture method. The manipulation of cultural conditions are very less, or it is almost negligible. Then optimized culture protocols. You will have a set of standard protocols and you have to follow that. So because of this optimized culture protocols that is followed, you will always have the same concentration of the constituents, and when you isolate, when you are trying to isolate, you will get it in huge number. Then unlimited supply of biomass. So unlimited supply means as you can grow maximum, and you can always throughout the year you can obtain. So these are the main plus points of ex situ aquaculture techniques. The opportunities that are available, of course, business is one opportunity. Another one is integrated aquaculture. So you can integrate, you can do genetic modifications, you can try for better quality drugs, all that in the growing stage itself, it can be carried out. Then valorization of organism with no commercial value. So those which are not having commercial values which are not sold. So those type of organisms can be avoided, and you can you can choose which organism you want to grow and develop. Then genetically genetical manipulation of the cultured organism. That is genetical modification can be carried out. And that is done mainly so that it will cure more number of diseases, especially for anti-cancer drugs, AIDS, and all such type of drugs which are quite uncontrollable by any other medicine. So for those, especially the marine drugs which are cultivated by ex situ aquaculture method are found to be very, very useful to the society. The weaknesses, information gaps. That is, you are having a method and you are growing it, but then the information that is received, that is, you know that the end product will be so and so, but then the intermediate products you do not know. So that is one thing, information gaps. Then high cost, of course, it is done under artificial conditions, so the cost is very high, and your manpower is also required. What are the threats? The threats are that there can be chemical synthesis because you are manipulating. So there can be chances of chemical synthesis. So the end product that you desire, you will not be able to get it. And here also diseases can occur, or any other new type of problem can take place when you're trying it for the first time, or if there is no proper environmental conditions which are maintained. So this is a picture you can see very clearly, students. All right, so the first one, that is A. First one you can see in white, it is written as A. So those are called as the spike cultures. And you can see how much it has grown, isn't it? You can see it, see such a huge bulk amount has grown. So A and B both are, B is a little bit more enlarged photograph, and you can see the width of it, it is in inches, and see you can get almost like 5 to 6 inches, you know, in length, and in breadth also almost it is like that way. So huge amount of culture can be carried out when it is done by this method. So this is it, it is in that huge artificial tank, and then the artificial conditions are supplied for it to grow. So the one is the spike method, and the other one is the vertical plates. So this is actually this green color that you see on in the diagram number C. That is completely algae, and on the algae you can see another type of fungi is also are also growing. All right, so this is a plate, and on the plate you are providing a base, and that base provides the nutritional requirements, and the microorganism is growing on it. So easily it can be scrapped off in pure form without the presence of any other types of mixtures, and then studies can be easily carried out, and it is easy to take it up and keep it, work on it, and then again place it back in the water so that it will grow more. Okay, so this is a diagrammatic view of it. Here, when we talk about development, what is the best practices when you are going for research, because my talk is mainly on how you go for research. All right, so you have to be well prepared. So first of all, designing as to what type of drug you want, and how you will prepare for it, and what type of experimental work can be carried out. So this is a design pattern which is required when you're carrying out research work, especially on marine drugs. Otherwise, you will get highly confused. So design and define what are the research questions. So like I have noted down in red, can you see www.marinebiotech.eu, then .marine biotech EU, marine biotechnology, ERNet. Like this, there are lots of websites which will give you picturestic view and information about the marine sources. All right, so the availability of it in our country, availability of it and elsewhere, and various things you can get from the source. So you have to properly assess it and set a protocol. You have to set the experimental protocol, which should be clear and crisp, and it should be attainable in the required time limit. Then compliance with ethical standards, because when you are doing on marine drugs, you have to acquire it from a resourceful person. Usually the government associations or the research centers are the best places where you can get the pure and the unmanipulated forms of drugs from there. Okay, and most important thing is checking whether it will provide a novel outcome. Novel means new. It should provide a novel outcome. So this is very important. Then you have to know about what is the experimental practice that when we carried out, what are the correct standards, the dose range, etc., and which type of constituent you prefer to isolate. Depending upon that, you can fix upon that, because it is not like plants wherein you have a lot of constituents. Usually they contain proteins or different varieties of microorganisms which are both gram positive as well as gram negative, and many marine drugs which are found to contain better activity than that of penicillin also. So studies are going on that. After that, proper drafting and manuscripting it, and then finally submitting the manuscript and patenting your studies. So now we are entering into the research part. So in college, what type of research or how you can carry out? So this is the general extraction procedure which can be carried out in labs by you. And in that, once you take your marine source, see the solubility of it, where in which it is soluble or it sustains. Okay, because usually you can freeze it, or you can make convert it into a state where it doesn't get destroyed. So usually freezing techniques are used. That from the literature review you can get to know a lot of ideas. Okay, so then it is first step is here it is extracted with hexane, that is the low polarity. So here it is the same thing. You can see it is the same way how we do the continuous extraction for the other crude drugs. So you have three polarities: the low polarity, medium polarity, and the high polarity techniques. And in low polarity, what type of constituents are isolated are like the fatty acids and acetogenins and terpenes. And in medium polarity, you get the peptides. Peptides are very useful, and you get small protein fractions also. Under high polarity is used mainly to isolate the alkaloids, amino acids, and the steroids, the big molecules and the saponins, etc. So like that it can be classified. So here a systematic technique I have given how you can carry out the extraction. You can see there are 3 to 4 stages of extraction wherein it is going from to different polarities. So this will be helpful for you all to understand how the exact extraction procedure is carried out. And this is a general procedure, the general procedure for extraction of marine drugs from the marine source. Am I audible? Hello?
Yes, ma'am.
I'm audible.
Yes,
right?
madam, audible.
Yeah, it's very clear, right?
Yes, ma'am.
Okay, fine. Now here I've taken one example of protein, because proteins, are very important. So, and they can be obtained even from the plants and animals and microorganisms of marine source also. So, proteins are usually, a lot of studies are carried out on proteins, and the peptides which are obtained from proteins are found to have a lot of activities and all the type of activities you can get in it. And even those who want to do major studies in anti-cancer can focus mainly on the peptide derivatives. Okay, So first step is, of course, this is a systematic study, like what are the different parameters, simple parameters, but this only gives a complete picture. This is the necessary thing which has to be done to get a complete project or a research, which we can say that, okay, fine, it is complete. So after you do the preliminary solvent extraction, then you can carry out a fractionation, or you can do the chromatographic methods of separation also. And once you get the crude protein isolated out, all right, the protein or the peptides, once it gets isolated out in crude form, then in order to get it more active, or to, this is a very good process, because you can carry out the fermentation of it or the enzyme assisted hydrolysis of it. So there are two techniques. And for fermentation, as you know, you can just keep it for some time, it gets fermented, or by using techniques wherein a little amount of glucose or yeast is added, so that fermentation process is mobilized, and that will give a different variety of microorganisms which are highly active. So that's why I have written that down. And after doing that, again, the preliminary extraction, ultrafiltration or gel filtration technique, and ultra centrifugation technique. These are the main techniques which can be carried out. Dialysis is difficult in the labs, so you can concentrate on the other methods. And then after that, the RPHPLC techniques can be used in order to separate and identify the constituents. Then further characterization using MS, NMR techniques, X-ray crystallographic techniques can be done. And then the peptides, once it is identified, then further even synthesizing it in the same type. If they are small peptides, you can also carry out further synthetic steps on it, right? And because then you will get a huge amount. That's why. Otherwise, you can't keep on isolating. So first, even you'll get a very small amount. It is very difficult for you to do the bioactive assay. So usually the synthesis is carried out after identifying. Otherwise, from that itself, the bioactive assays are performed. Bioactive assays are almost similar to that of the standard assays which are carried out. Only the extraction process is little difficult, and the assessment methods. So these are the various steps involved, which has been charted out mainly for the marine source. Now, why we are focusing? So in India, there are around 8,000 kilometers of coastline, and Kerala especially is in the coastline itself. So there are many research centers in Kerala, which people are not aware of. So government itself has started so much. I've just listed out a few here. You have the CSIR, NIO. NIO stands for National Institute of Oceanography. Okay, so it carries out a lot of research on marine drugs, and also they call over, or they take, if you are sending any material to them, they can carry out the studies and give it to you also. It is situated in Kochi. Then you have the Center of Marine Living Resources and Ecology under the Ministry of Earth Sciences, which is also in Cochin. Then Central Marine Fisheries Research Institute, third one in Cochin, and Calicut Research Center, Calicut, which is also situated in Kerala. In Karnataka, Karwar district, you are having the Central Marine Fisheries Research Institute, which is doing an immensely wonderful work on marine research. Now here I've listed out some institutions or universities which are giving training. So if there are any B.Pharm students available here and they would like to, after that, persuade into research on specific areas, if they are interested in marine drugs. So this is the Channeling You can directly try to get through over here. So in India, I've listed out some important ones. That is the National Institute of Oceanology, which is situated in Goa, Cochin, Mumbai, and Visakhapatnam. And next one is the Central Salt and Marine Chemicals Research Institute, which is in Gujarat. Central Drug Research Institute in Lucknow. So here, these are the institutions which offer training only for marine research. Okay, and on the right hand side, on my right hand side, you can see the names of the courses. Even though it is written as MSc, it's okay. You have PhDs also in all these centers. All right, so these are just because I can't write like MPharm and all. So, but still, after BPharm or a PhD also can be carried out in these places. Now, this is outside India. There are outside India, there are many centers, but the most important, those who are doing the work on marine drugs, which are highly highlighted, that only I've listed down here. And do you know marine drugs research was first originated in China and Japan. And there are a lot of marketed drugs available, though they are only as nutraceuticals and cosmetics. There also, but still a lot of drugs are marketed from marine origin. And the first countries which researched on them are China and Japan. So these are the list. The Roche Research Institute of Marine Pharmacology, which is in New South Wales, Australia. Then the Marine Biotechnology Center, University of California, USA. Ballonese Lab, which is in Connecticut, USA. Okay, so these are the programs which they have, and they also have a lot of PhDs. You can register if you are interested in marine research. Now, here some recent outbreaks for the discovery of potential marine drugs. So in the first one, you can see FDA protein ligand interaction. All right. Now, I have divided into 4 parts. In the fourth one, now, in order to understand before, this is about docking. All right. Now, why? First, I'll tell you why docking is important. Molecular docking means you don't have to really carry out the synthesis, or you don't have to really carry out the process of bioassays. It is all done by computer aided drug design technique. Okay, molecular, it is computer CADD. It is done mainly for marine drugs, because the reason why this is a really upcoming field is because it takes around 10 to 20 years for a drug to come out, for a drug to come in the clinical trials and then to be marketed and to be cleared by FDA. So computer aided drug design, once you know the structure clearly and you know the ligand. So the ligand is the base and the protein. The protein is the one that causes the effect. So, you know, in the case of COVID virus also, the same technique wherein the spikes were identified and the structures which had the same spikes were mimicked, so that the coronavirus will get confused and then there will be replication or the breaking of it. So, or the death of the coronavirus atom. So that is the reason why molecular docking is very important. And it is like a lock and key mechanism. Okay. So once you know the structure, and since it is in the computer, all the properties, all the pluses and the minuses of binding, everything is computerized. There can be no errors by human beings, and there are no environmental factors, no time loss, and it is found out to be very, very perfect. So once you come to know this, then synthesizing the drug is also very, very easy. Okay. So FDA protein ligand interaction. So FDA has found out like this way. So below that in red, what I've written is cytarabine, DNA polymerase, vidarabine, adenylyl cyclase, then eribulin, tubulin complexes. So these are the protein ligand interactions, and they are FDA approved, wherein you can develop even more and more drugs, marine drugs, which are having very high potency compared to that of the drugs which are available here. And because of bacterial resistance and new mutations of the of the microorganisms that are taking place because of the low immunity levels of people, the lifestyle problems, it is very important that natural drugs, or which will mimic, which will be same like that of the naturally or obtained compounds are synthesized, because only then you can be sure that okay, they will not harm the system. So that's why. But there are also challenges. The challenges are that it is complicated. Some of the molecules are very complicated, and it can cause multi-targeted components. All right, so multi-targeting will be there, due to which there needs to be a expert to do these studies, a real good expert. So this is an area, molecular docking, which is highly coming up in the field of research. So molecular docking, it predicts the binding mode and the binding energy of the protein ligand complex. All right, so it predicts, it's like a prediction. So once it predicts, and yes, it can join in, and then once the protein ligand complex is formed, you can come to know that it can destruct the attack by the other microorganisms or the viruses. Designing and optimizing the drug lead compounds. So you need to have, now this is the era when everybody is focusing on lead compounds. And in order to design, you can design. So once you know the structure of the ligand, all right, then you can design your own lead compound, and that where it is available or where you can obtain it, that you can easily find out. And in marine drugs, you have multiple variety of components. Like each marine source will have thousands and millions of active bioactive constituents, which are of various molecular weight and molecular formulas, and therefore obtaining it from the marine source is quite easy. And once you know the activity of it, it becomes easier to carry out the molecular docking. Now here there is a picturestic view. You can see here in the diagram below, right, you have the A and the B diagram. So here the ligand, it is a biomolecular protein. It can be an RNA or it can be a DNA. So that I have given it in the color over here, a different color, which is the red color. And then interactive regions of the surface. So which is the interactive region which causes where it can attack, so that we can find out whether it can go and fix itself like a lock and key mechanism. All right, now you can see that receptor protein, which is shown in the green color, that is the receptor protein. And these proteins which are present, like if it is the lung cell or the liver cells or heart or anything where you want specific targeted action to take place. All right, or anti-cancer where you want the apoptosis action. So whichever action, you can design it accordingly, and then depending upon that structure, if the compound is having the same activity, it can go and fix to it. All right, and for this there are various types of algorithms which are there, which is called as the FTT fast Fourier transform, the ClusFLO, ZDoc, DOT. These are all the computer docking big instruments, or you can say the computers which are highly sophisticated. It is like artificial intelligence which can cause the simulation studies. Right. Now, the target proteins. The target proteins, you can see here some of the studies which have been carried out and as we have successfully obtained. One is in that melanin formation has been successfully carried out by molecular docking methods. Then for diabetes, for diabetic mellitus, also drugs were found out. Hypertension drugs have been found out, anti-inflammatory drug, cancer, anti-cancer drug, and then the SARS-CoV virus 2. Against that also now recently antiviral effect or anti-COVID effect drug has been isolated or found out from by molecular docking method. Otherwise, it will take a lot of time to analyze, but it has been studied that the fluorotannins, the oligomers, then which are obtained from the Sargassum spinolegerum brown algae is found to have the anti-COVID activity. And it is now the most recent publication which has come, and a great step ahead in the field of natural drugs. So this is it, a little bit in elaboration, how the studies you will get. So you can see here the structure of the ligand, it is given on this side, and all the colors, that is the sites which are given, they are all the groups and how the binding takes place. So this is a computerized way, what you can, how it is fed in the computer. So the binding can be easily studied, when you know the sites and finding out which type of compound can be used to form the computer protein ligand. Now on top, you can see one graphical diagram. All right, so it is on the x-axis. You have the one of the marine source, that is the firefly luciferase, and that is the percentage, that is how much is their concentration, and down you have the studies as to what type of activity, and you can see the range. It is compared with the other standard drugs, and one is you can see in for SARS-CoV-2 studies you can get very good effect. It is better than that of the with the standard. So it's comparatively very good antiviral effect has been found out. Now here I'm going to take you through a journey of comprehensive marine natural product database. Now this is a database. Now everybody will be thinking that it is so difficult to find out, to find the literature and then to remember the complicated names. So for that, nowadays a lot of database is provided mainly on marine natural products. In short, it is also called as MNPs. All right, marine natural products databases are available, and I'm going to take you through the journey of a free database, and the name of it is the CMNPD database. Okay, so what is CMNP? It is integrated platform for structure dereplication for discovery of new lead compounds. You see, means it is mainly designed for discovery. It is mainly designed only for enhancing the research in marine drugs. And those who have found out this or collected the database have made it free for everybody. It has more than 31,000 chemical entities. Very easy to access and find out. And various physicochemical and pharmacokinetic properties, everything is listed in it. what are the standardized biochemical, biological activities that have been performed, on which it has not been performed, which is in pipeline, and where you can focus. So you can easily find out, because it is totally updated. It goes on updating. So as it has been, it is a continuous updation. So the latest information you will get from CMNP. The taxonomy of each marine source, the geographical distribution of the source, which area where it is available, and then the literature citations also. You can get the complete literature citation. So something like this. Below you can see 2 circles. I will elaborate and show it. I will be taking you to a visit to the site in my next slide. Okay, so something like this. I hope you have understood this. CMNPD, it will be very helpful. Have you all understood what I'm saying?
Yes, ma'am.
Yeah, okay. Thank you. Right. So here I'm going to enter. Okay, can you see the website? All of you? Are you all able to visible?
Yes, madam. Yes, madam. Visible.
Visible, no? Okay. So here you can see, this is the home page, right? There is one quick search button over here also. So if you want to type in any name, family name, everything that will be open. I'll take from here the browse button, and then we come to compounds. And now you can see all these compounds are obtained from the Animalia species. And you can see here the taxonomy. This is 19,773 compounds are there, with the structure, the name, structure and the name. Everything is available here. Fine. So molecular docking, carrying out research, everything will becomes very easy. So you have for all, the next one like Archaea, there are only 3. Fine, there are only 3. So here also you can get the molecular structure and the names. Fine. So you can see for fungi, you open like this way, and then there are 20 pages. You can keep on scrolling, it will come down. It's taking little time to open. All right, so like this way. And now imagine I selected this. Fine. Now I click on this and I get the database of this compound. See, getting the name, the molecular formula, the molecular weight, the UPAC name, stereochemistry. Fine. Everything. Yeah. Just see the amount of database which is available, even the activity, the solubility, the binding model, right? So full thing you can get like that. Now we come to documents. You want to check any documents, you can see how many patents. If you want to know about patents, all these are the IDs. So if you, and the journal name, everything will come. So you have to just click on one, on which year the patency was taken, everything you'll come to. 1977 publication year, aspects of marine drug chemistry, who is the author. Right. And below, when you come, you can check here the molecular weight. So this marine source has got various compounds in which molecular weight of 100, it is so much, concentration is only so much, but up to 200, 250, you can see around 40% is there. All right, 300, but then you have of low molecular weight also. So very low molecular weight. So these are the families, different types, which are in which you have the quantity, right? Like this way, everything will be available just on the click of your fingertips. Right, now we come to here, visualization. Now, when you come for visualization, this is the ring that I was telling. This is the, I should say, this is the bibliography of all marine drugs and plants which are available. Now, this is very interesting. Now, you see, this is a big ring. It's like a full chakra, and you want to check on fungi. All right, you can see there are how many databases? There are 4,683 fungi databases they are having. Now these names, all these names you can check on. So I click on fungi. Right, so fungi has come in the center. Fine. And these are all the families. Now if I click on any one, the ring goes on and on. So, okay. So you see the species name has come. All the species. Right. So this is having only one species. That was the end. Right. Back. Right. So this is about visualization where you can check in everything just on the click of a button, and whichever, see, this is the last one, after that is on there. So you can see if you want to see on bacteria or porifera. Porifera is a very good marine drug. Very interesting. So there is no need, this database will give you a clear picture, and whichever you are selecting, other synonyms of it, availability of it. Now I'll show you how you can see the availability of it. Now here in this you can see draw structure, structural classification, click to draw query structure. If you have a question, you can click here and you will get the answer from them. And it is all totally free. Here you can give the name and then you can get the answers. And if anybody has found out anything new, that also can be, can be added on to this. Now here you can see, if you take any one type, what type of targets, where it is targeted, how you can get the name of the organism, how many compounds are found out, how many are bioactive. All right, so everything you can find out from this, and what type it is. It's a single protein. You can see these are all single proteins. So like this way, totally you can get data from the CMNPD. There are many other databases also which are available, but many are paid. It is all, many are paid. This is being given by the Peking University. Peking University carries out in anti-malarial studies, and you can see over here data in brief, target name, assay type, activity. So which is not having the activity, you want to do research or you want to carry out any new findings, by within 3 or 4 days you can come to know everything. You can do the selections. All right. Bioactives, you can see there are 72K, and those who want to synthesize also, it will be easy because the complete bioactivity, the structure of the molecule, everything will be obtained. And documents contains, this is fully literature, all citations. Documents means you will get the complete literature about that particular marine drug. Full literature can be obtained. Okay, so this is about it. So I hope it was little bit interesting. Can I get a yes or a no?
Yes, madam.
It was interesting, right? Good. Happy on that. I'll start with the slide. All right, so this is the visit that we have gone through. Very wonderful database it is. Now, what are the future prospects? So future prospects is production of biomass. So whatever I have told you earlier, that only future prospects and summary, because let us not think of the problems, and the challenges will always be there, but it has been overcome. And there are many methods in which many areas where you can get help and try to carry out good studies. So the work which has been carried out, or the promotion that has been given to develop drugs from marine source, is very huge. And throughout the world, scientists are now, they have plunged into this field of marine drugs. The saturation point, we have reached the completely saturation point of all the drugs which are obtained from plants. And you know that there is nothing like this way that you can get so much of data or so much of CADDs. All these are not obtained when you're doing the work on plants. So you have to go for the latest, and marine drug research is of course a area where you can really achieve something, and you will feel that your research has been very useful to the society and mankind. So we, the pharmacognoscists, have to focus more on the latest, and which is most important. Even if you want to synthesize the drug, you find out what is the basic structure, and then further synthesis also carried out. You know the basic structure, and then from that synthesis also is possible. So you know how it can be developed and patenting it, because immediate patenting is possible. And as soon as you have carried out any, or you have found out a lead molecule or a rare molecule or a new molecule, novel molecule from the marine source. Right. So this is all that I would like to share with you all. And thank you so much for giving me this opportunity. And I hope all of you have enjoyed as much as I have enjoyed sharing it with you all. Fine. I've kept the level of researchers and students, so I've tried to make it feasible for all. So I hope it was useful. Thank you very much.
Thank you, madam. Thank you so much, madam, for your highly informative session on marine drug research. And you covered all the aspects related to this marine drug research topic. You starting from sources, isolation, identification. You also covered molecular docking, and CMNP database also introduced for us. Thank you so much, madam. In the chat box, there are no questions. Now I request Dr. Raghava Kalyan to propose vote of thanks.
Thank you so much, sir.
Thank you, madam.
Thank you, sir. So we came to the end of the session, and once again, I would like to congratulate, and I wholeheartedly thank the speaker for providing us the most widest platform that one can ever have on this pharmacognosy thing, because especially the docking phenomenon, and especially the software that she introduced to us, is something that is very exciting, and I think the people who are actively involved in research can get good benefits out of it. So I once again congratulate her for putting her information on this platform so that everybody can be bestowed out of it. And right now, I would like to conclude this webinar with a very short vote of thanks. At the outset, on behalf of our institute, a very big thanks to the speaker, Dr. Sapna Srikumar, for accepting our invitation, and also for giving us the most widest possible platform where we can explore the research pertaining to marine research. Ma'am, thank you once again. And next, I would like to thank the management of Chebrolu Hanumaya Institute of Pharmaceutical Sciences for giving us this wonderful occasion where we can reach to different professionals in the field of pharmacy so that we can bring everybody together for better research, especially in a times like this where our capabilities to interact directly is limited, so we can carry on with these programs. Next, I also thank the principal of our institute, Dr. S. Vidyadhara, for not only encouraging us, but also for leading us to find eminent personalities all over the field of pharmacy and bringing them on this platform. And I also congratulate the Department of Pharmacognosy and Phytochemistry for successful handling of this webinar. And last but not least, I thank all the teaching and non-teaching members of this institute for their active participation so that this webinar can be conducted in a very smooth manner. I thank all the participants for your active participation, and we always promise you that we bring the best for you every time possible. Thank you once again. Goodbye, everybody.
Thank you very much. Thank you, Dr. P. Vijita. Thank you, sir. Thank you to Vidyadhar sir and everybody.